F1 hybrids. Let's talk about mice. Fun fact about mice. Mice, as you all know, have 20 pairs of of chromosomes. They have 20 pairs of chromosomes. This is, of course, in sharp contrast to humans that have 23. Humans have 23 pairs of chromosomes. This is not the only reason why mice are studied as human models for medicine and a whole bunch of other things. But, you know, it doesn't hurt, that's for sure. Each chromosome is a big string of genetic information. And it's a big string that's sort of woven around a bunch of pieces of protein in order to make it much more compact. It's a big string of genetic information. Often, we use just a rectangle, we use just a simple representation of DNA, of the chromosome, in order to make our life easier. And one of the characteristics that's going to matter about chromosomes is that you have a particular piece of information on a particular chromosome. It's going to turn out that we can find that out for particular things in remarkably easy ways, you know, without actually having to go in and read exactly what the genetic code is. So we will often use just a rectangle to represent a chromosome. Like so. Beep, 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 beep. Rectangle. That's a smiley face looking at the rectangle being like, yay, all good. So here we go. If I were to draw the human chromosomes, I could use the size of the rectangle to represent the length of the chromosome in base pairs. Base pairs is a concept that we will encounter later. It will make a lot of sense. So, if I were to do that, then you would find that the first chromosome would be nice and long, and the second chromosome would also be nice and long. The third chromosome would be substantially less long, and then the fourth chromosome would be less long again, and on and on and on. 1, 2, 3, 4. You would find this for all 22 chromosomes. In fact, they are ordered quite naturally by length. 1, 2, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, and of course, 22. The last two, however, are totally different. In fact, if we were to organize the X properly, it would be one of the larger chromosomes again. This is our X chromosome. They should all be roughly the same width, I'm sorry. But our Y chromosome is just a titchy little guy, about half size. Y. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, oh man, have I done it right? 21, 22, X and Y. Now, the X and Y, the last two, are special, and they are called the sex chromosomes. The sex chromosomes differ quite a lot depending on the species, and there's a whole bunch of different variations of them that you can go look into yourselves. Birds, I think, are X and Z, or is it Y and Z? I think it's Y and Z. Very interesting stuff. What the XX and the XY chromosomes is referred to in biology as the biological sex. It determines biological sex. So in this case, this would be not male but female, and XY over here, just like that, would be a biological male. In a humans and some other mammal species as well. So this particular guy, XX, is just the presence of 2 of these X chromosomes, and this particular guy, XY, has an X and a Y chromosome. Very important basic difference on the chromosomal level. From there, that's where we get the different genetic expressions that lead to things like differences in puberty, differences, basic biological differences across the board are seen to be attributed to sex chromosomes, and the science of actually tracking down exactly what trait is responsible for on what particular part of which chromosome is advanced and complicated. Yeah, and that's essentially what we're starting to break apart right now. For us, however, we're much more interested in the mouse and the mouse chromosome because we can do studies on mice that they won't let us do on people for simple reasons. So, numbered by size, these guys are all numbered by size, with sex chromosomes last. With sex chromosomes last. Humans are diploid. Which means that chromosomes are paired up with a similar copy later on. So this is still a lot of things to draw, even in our simplified form. So one of the ways that we're going to simplify chromosomes down even further is we're just going to draw a couple pairs. So the pairs keep the diploid nature, as our mice, present inside of each cell. So I can draw, for example, these guys like that, and that makes it clear that these are shared, or it should hopefully try to make it clear that these are shared pairs of chromosomes. All right, so this is a pair, pair, 1 pair, 2 pair, 3 pair. Now in this particular case, I don't want to use this for humans, I want to use it for mice. And we're gonna give our mouse a silly name for now that I'll explain a little later. All right, this is P-Man. Welcome to P-Man. This is P-Man. P-Man is having a pretty good day. Such a good day, in fact, the P-Man is about to get crossed with another mouse, which means more baby mices in the future, and P-Man was about to get crossed with P-Paul. Lucky day for both mice. P-Paul also has chromosomes. Thank goodness for them. And involves my first use of color. Is this actually going to work out? Oh my gosh, look at that. It's beautiful. So the colors here just indicate that we've got a different set of genetic material for each set of mice. The P-Man species has all the same sort of material, and the P-Paul species has all the same sort of material. What's interesting is what happens when we cross P-Man with P-Paul. P-Paul. When we cross P-Man with P-Paul, something interesting happens. Their chromosomes mix in a predictable way. The chromosomes mix in a predictable way. Basically, one copy from each parent. One copy from each parent. This generation also gets a name. This generation is called, I used the purple. F1 hybrids. Now, technically, the name would be F1 hybrids of P-Man and P-Paul. F1 hybrids of P-Man and P-Paul. One copy from each parent, nice and straightforward then. We need one chromosome from this guy, one chromosome from the other guy, one chromosome from P man, one chromosome from P Paul, one chromosome from P man, one chromosome from P Paul. All right, now they all look like they're from P man, so let's make this one look like it's from P Paul. So this one looks like it's from P Paul. And last but not least, this one gets the P Paul treatment. This is then the basic chromosomal genetics of this hybrid, which is kind of cool. We figured it out. Note: mice perform meiosis before they hand on their chromosomes. As we just talked about. But for the characteristics we're looking at, they're the same within the species. The characteristics we're looking at are the same within the species, within each species. So we can ignore that hybridization, we can ignore that randomization of the chromosomes during that F1 hybrid stage. It'll come back later though. Hope that argument makes sense. Basically, the idea is just because each line is, we're looking at traits that are maintained across each line, then we don't have to worry about the randomization of the traits inside of their own group. Does that kind of make sense? This is kind of why F1 hybrids are so cool and important. If we're doing relatively substantial crosses, it allows us to sort of have a control set of genetics on either side. We're about to mess with the genetics. We want to see what happens when we change things. That's always the trick of biology. So we're then going to continue with another cross. We just did a first cross. Just did our first cross with mice. And of course, we're flush with power, so we want to do it again. Let's take our F1 hybrids this time. These guys, with the one set of purple and the one set of regular, empty looking chromosomes. Like so. 2, 3, bang, bang, bang. These are our F1 hybrids. We're now going to do something that's a little crazy. We're going to do a back cross. Ooh, a back cross. Now, a back cross involves taking our F1 hybrids and essentially breeding it with one of their parents. Terrifying, terrifying. So away we go. We're going to do a back cross with, which one is it again? It's P-Man. Yes. So P-Man, of course. P-Man has our empty. Guys, just like that. And now, what's our result? Well, here, meiosis occurs. Meiosis, meiosis. See, one of my deep fears is that I mess up meiosis versus mitosis. But I'm right, still meiosis. Meiosis occurs, sexual reproduction. I know I can do sexual versus asexual, but meiosis versus mitosis, it starts to blur for me, guys. Meiosis occurs mixing the chromosomes together. Of the F1 hybrid. So their chromosomes get mixed, but by the same logic, P-mans get mixed, but don't have any effect on the traits that we're looking at, so we can just draw those ones straight up, straight as is. So I'm going to do the donation now from on the right-hand side, just as I did before. And on the left-hand side, what we get is this mixture. I'm going to toss in a couple places there. So it looks something like that. That actually, there we go. Sorry, I'm not sure that one was actually possible. But there we go. You get your rough idea. Let's do a bigger one over here. Nice. So here we have the randomization from the meiosis over here. So meiosis means that the contribution is randomized from each of the F1 hybrids. Whereas the fact that we're dealing again with the meiosis between these two similar lines means we don't have to worry about it when we're putting in the other side of the P-man gene. Wow. So what we get here, these guys are back-cross hybrids. And what we discover is that these actually have four different unique behavioral types. These back-cross hybrids have four unique behavioral types, which means that the mice actually show demonstrable different behaviors based on this simple genetics that we can do on a piece of paper, which I think is really cool. Have four unique behavioral characteristics that we'll talk about next time. So thank you so much for watching. I thank you so much for being here. I thank you for enjoying yourself and having a good time and just being a good person in general. And I will see you next time. Bye for now.